Hi everyone, welcome to Medical Microbiology. So today we will continue chapter 12. We will discuss about the antibiotics, which is drugs that use for bacterial infection. Okay, so this is type of antibacterial. Okay, just remember, okay, so what we call the antimicrobial drugs. So this is a general term for drug that used for any type of microbial infection. It can be divided into several groups. The drug that only used for bacteria is called the antibacterial drugs. Okay? So this is antibiotics. So remember, antibiotic is for bacterial infection. If it is natural, production is produced by nature, then it's just called antibiotics. But if it is made in the laboratory, then this antibacterial drug is called as a synthetic drugs. Okay? And this is the one that we will discuss in this lecture, okay, lecture number 3. And then if it is used for fungi, then it is called the antifungal drugs. If it is used for protozoa, then it is called the antiprotozoal drugs. If it is used for virus, then it is called the antiviral drugs. This is actually one more, but it will not be discussed in chapter 12, which is anti-helminthics, which is worms. Okay, anti-helminthic drugs, which is for worms infection. Okay, so in this lecture, lecture number 3, we will discuss these antibacterial drugs. Then we will discuss these 3 type of antimicrobial drugs in lecture number 4. Okay, so antibacterial drugs or antibiotics. The first one developed is actually the penicillin. And then after that, the drugs or the antibiotics are becoming more and more and more. There are some natural products, and there are also some drugs or antibiotics is produced in the pharmaceutical lab. So this penicillin is actually produced naturally by fungi, which is in the species of Penicillium chrysogenum. So this is the major source for natural penicillin. So this is the list of the penicillin drugs from the first type, which is natural type, penicillin G. So this is naturally made by Penicillium fungi, and then penicillin V. So this the natural type. It is made by Penicillium. And then after that, some drug is become semi-synthetic. Some is actually produced in nature, and then there are some modification in the lab. It's become semi-synthetic drug. And finally, the last generation, there are some drug that especially made in the lab, which is synthetic type of penicillin or antibiotics. Okay, so these drugs, the one that destroy the cell wall, mostly they have the same nucleus structure or center structure. Is called the beta-lactam structure. So all the drugs that used for destroying bacterial cell wall, they usually have this beta-lactam structure. That's the main structure for this type of antibiotics or drugs. So this is this is the nucleus of the main structure over here. Okay, so the beta-lactam structure. The difference between the drugs, different between the penicillin, cephalosporin, amoxicillin, for example, usually on this, what we call the R group or side group or radical group. So this make different type of antibiotics, different type of effectiveness, whether broad spectrum or narrow spectrum, and etc. So the pharmaceutical company usually playing in this area over here on the radical group. So they change the radical groups in order to produce maybe more effective or broad spectrum type of antibiotics. Again, penicillin G and V, this is the most important natural form. So this is made in nature. And then there are development of semi-synthetic penicillin, which is some part is made by nature, and then in the lab they put some additional structure, especially radical structure, in order to produce more broad spectrum drug. And then there are also some antibiotics that all the process is made in the lab, which is the synthetic drugs. The same thing with the cephalosporins. So initially it is made in nature by this fungi, okay, Cephalosporium acromonium. So this is fungi that produce this cephalosporin. And then the first generation usually natural made cephalosporin. Then in the lab, they're creating another generation, like second generation, third generation, and fourth generation that is created in the lab. And this new generation usually more broad spectrum type, so widest range of use for different type of infection. Again, the way they made that second, third, and fourth generation is just playing with the R group over here, radical group, which is radical group number 1 or radical group number 2 or side group number 2. The nucleus or the center is gonna be the same. They all have the beta-lactam structure over here. So this is beta-lactam structure. Okay, so they just modify these radical groups in the lab, in order to create a new generation of cephalosporin that is better for curing wide type of infection, maybe gram-positive and also gram-negative bacterial infection. Additional beta-lactam drug like carbapenems, monobactam, this is also used for mostly for gram-positive bacteria, because gram-positive bacteria, they have the thicker cell wall that usually is become the target for these beta-lactam drugs. And again, remember, gram-negative bacteria, their cell wall is actually covered by additional membrane, which is a protein or lipoprotein membrane that sometimes protect them from this type of cell wall destroyer. So usually the one that is used for cell wall destruction, mostly more effective against gram-positive bacteria. But again, the broad spectrum type of penicillin, cephalosporin, they also able to destroy the cell wall of gram-negative bacteria. There are also some non-beta-lactam cell wall inhibitors. Okay? So like, for example, bacitracin. These drugs or these antibiotics do not have the beta-lactam structure, but they also able to destroy the cell wall. Mostly, this bacitracin is used as a topical agent or topical medicine or ointment. There are another one is isoniazid or INH, mostly used for bacteria that actually cannot be classified as a gram bacteria, whether positive or negative, like Mycobacterium tuberculosis. Remember this Mycobacterium TB, it has cell wall, but the cell walls, cannot be really tell whether it's special cell wall is different than cell wall of gram-positive bacteria and gram-negative bacteria. So it has different type of cell wall. I think that's all for antibacterial drugs. And next lecture, lecture number 4, we'll talk about the antifungal, antiparasitic, which is antiprotozoal, and antiviral drugs. See you on the next lecture. Bye.